I'm David Lakomy, a second-year fellow with the Choose Development program from the Society of Developmental Biology, and this is our fish room in Dr. Wagner's lab. And here is our lab. So, here are our hatcheries where we cultivate Artemia to feed to the fish. Here is our benches where we do most of our lab work. And here is our injection room where we inject embryos. It's my favorite room because injecting is like playing a video game and I really enjoy it. And then the last room is our microscope room where we do all of our imaging. So in this room we have a stereoscope and a compact scope. My first semester here I took a freshman seminar in which we read papers written by Rice faculty, and one of the papers was written by Dr. Wagner. And after reading and analyzing the paper, we got to visit the lab, and in the lab I just realized I was actually really interested in the work that they were doing here and I wanted to be a part of it. So the next semester, spring of my freshman year, I officially joined the lab.
I'm Dan Wagner. I'm an associate professor at Rice University, and David Lakomy has been working in my lab since he was a freshman. My lab is interested in using zebrafish to understand the genetics of early vertebrate development. Historically, we've worked on early development, and we've begun to branch out into cell differentiation and organogenesis, and it's these kinds of projects that David's actually been pioneering in our lab. In particular, he's been working on developing a model for port wine stain, which is particularly challenging. Recent work has shown that somatic mutations lead to this very common congenital vascular malformation. And modeling that in other organisms is challenging because it's relatively infrequent. So David's been developing some innovative tools that will allow us to create a model for port wine stain that will be much more robust and allow us to look at exactly what's going on in this vascular overgrowth phenotype.
And although we've tried different things in the past, we are focused on trying to create a Cree-inducible line of transgenic port wine stain fish to be used as a model. And hopefully this model, if useful, can help elucidate different pathway components and different mechanisms of how this mutation is formed and manifests itself, and hopefully in the future can provide some insight into some potential drug therapies.
Hi, I'm Wagner. I'm a second-year grad student and I've been in this lab for about a year now. I'm working on the P38 pathway. It's a stress response pathway, so whenever our cells feel any stress like heat or hypoxia or osmotic shock, this pathway is activated and it activates a number of different processes.
And lastly, I just wanted to express my thanks to the Society of Developmental Biology for helping fund this research for me over the summer, as it is an integral part of my density here at Rice, and I gain a lot of value out of it. And I am extremely excited to be able to actually express that and share that value with others at this Seattle meeting. And finally, I just wanted to give my final thanks to the Wagner Lab for all these years, having been extremely supportive and very educational, and just a really lovely experience overall. So.